Our first speaker is Pedro Baldoni from WEHI. He's a postdoc in Gordon Smythe's lab, and he's going to tell us about quantifying transcript uncertainty using edgeR. Welcome, Pedro.
Blink green. Can everyone hear me? Okay, I think it's on. Okay. All right. So I'm Pedro. I'm postdoc in the Smythe lab at the WEHI, and I'm really excited to present today this project that I've been working on performing differential expression analysis at the transcript level with edgeR, and share some of our ideas with you how you can analyze your RNA-seq data at the transcript level in edgeR. Okay. I'll do it. It's not going through. Should I ask? Okay, cool. Thank you. So I'd like to begin my presentation by, first of all, contrasting transcript level analysis with the type of analysis that people have been mostly using in the past 14 years of RNA-seq, which is gene level analysis. So in gene level analysis, you start off with your sequencing reads, and you select your favorite alignment tools, such as, for example, R-sub-read. You're going to map those reads back to your reference genome, count reads on features, in this case, genes, and perform your downstream gene expression analysis. But in the past 6 to 7 years, with the development of Salmon and Kallisto, these two quantification tools are making the analysis at the transcript level more and more common. And that's because Salmon and Kallisto are mapping reads back to a reference transcriptome, hence making the analysis much more, providing users a much faster alternative than the standard aligners for gene level that you use for gene level. But there's a price we pay there. So as I show here in this diagram, we have certain reads that map ambiguously to certain transcripts of that gene that we want to study. And that's because there is a high level of sequence homology among transcripts in your annotation. When you try to summarize those data into a matrix of assignments of reads to transcripts, we have lots of uncertainty there. And when we think about uncertainty, we think about missing data, we think about extra variation that will be carried to our downstream analysis. So if you ever attempted to perform a differential expression analysis with edgeR at the transcript level, you remember that the expression estimates that you get from Salmon and Kallisto are wildly variable. What I'm showing here is the biological coefficient of variation from edgeR, first at the gene level on the left, and then transcript level on the right. We can immediately see that the estimates at transcript level are much more variable than the gene level. For transcripts that overlap with each other, we have overestimated biological variation, and then we see those clouds of points at the top left. It's very typical for a transcript level analysis. We can also look at the mean variance relationship, both at the gene level and transcript level, and the well-established variance function that people assume for gene level analysis, which is the negative binomial. We can see that that variance function doesn't fit well anymore at the transcript level data. So we really need to think about these sorts of variation and try to account that properly in our statistical framework. So let's go back to our experimental design and try to assess where mapping ambiguity and these uncertainties are arising and how we can account that in our framework. So suppose we have our 4 different mice for which we want to run an RNA-seq experiment, and we have a certain gene that we want to perform a gene level analysis. We have a relative abundance of that gene that we do not observe, but we select fragments and sequencing reads from that gene that we compute read counts then. When we talk about biological variation, we talk about this variation across biological samples, and in gene level analysis, the technical variation that you get is pretty much from that sampling of fragments and mapping reads back to a reference genome. At the transcript level, however, this story is very different. We still have the biological variation, but now we have a huge mapping ambiguity when mapping reads back to a reference transcriptome. And the Poisson variation that we usually see in gene level analysis is no longer Poisson, but actually a combination of the variability of the Poisson sampling and the mapping ambiguity of assigning reads to transcripts. So we did an experiment, very simple, to simulate data with technical replicates to assess what type of variation is coming from this mapping ambiguity. So we generate technical replicates, which supposedly should give us Poisson variation. And what I'm showing in this slide is the mean variance plots from the true counts at the top and the counts that you get out of Salmon at the bottom. Each point in that plot is a transcript, and I'm coloring with respect to the number of transcripts being expressed by the genes associated to each transcript. So if a transcript is coming from a gene that only expresses a single transcript, that point is red. Otherwise, if the transcript is coming from a multi-transcript gene, that point is black. Immediately what we see is that for transcripts that are coming from multi-transcript genes, the variance that we see from Salmon counts is no longer equal to a Poisson variation because we don't see those clouds of points along the diagonal anymore. So to take care of that process, we propose to use a quasi-Poisson variance model to allow the variance of those transcripts that are overlapping with each other to be greater than the mean. So ideally, that factor that we have now, sigma square, which we call mapping ambiguity over dispersion parameter, will be equal to 1 for the transcripts that do not overlap with each other. And for transcripts that are overlapping with each other, we allow that sigma square, the mapping ambiguity over dispersion, to be greater than 1. The issue with that is that that causes many problems downstream in our statistical framework. First of all, because sigma square, or mapping ambiguity over dispersion, depends on the topology of a transcript in your notation. And the variance function that's marginal to the abundance that we really care about now is very transcript specific, and of course, no longer a negative binomial variance that we usually assume in gene level analysis. So in edgeR, to deal with that, we propose a count scaling approach. So we propose this approach where we scale counts by the mapping ambiguity over dispersion. That allows us not only to compute in closed forms expected values of those counts, but also to write down the variance function in closed forms. And you may notice that now we have effective counts that reflects the true precision of those transcripts wise estimates. And the variance function is, again, negative binomial, which suggests us that we can use methods developed for gene level analysis to data coming from transcripts that are properly transformed, accounting for the ambiguity. But how can we estimate sigma square? Because it depends on how these transcripts are overlapping with each other. So to do that, we use Salmon and Kallisto. Salmon and Kallisto will compute for you Bootstrap estimates at the level of equivalence classes. So in equivalence classes, we have sets of transcripts for which each read can map unambiguously to. And because Bootstrap is done at the level of equivalence classes, we have some sort of replication of that uncertainty, because for each one of those Bootstrap samples, Salmon and Kallisto will run the probabilistic assignment of reads to transcripts over and over again. So the key idea here is to estimate the mapping ambiguity over dispersion and quantify that extra uncertainty that we have to the Poisson variation and estimate that ambiguity using Pearson residuals, for example. So in edgeR, we estimate that mapping ambiguity, and the only thing you need to do now is to scale down your count by the ambiguity associated to those transcripts. And you can perform your downstream analysis as usual, for example, with the quasi-likelihood pipeline. So the workflow to perform a differential transcript expression analysis with edgeR is quite straightforward. We can start off with your FASTQ files of the libraries that you want to analyze. Your transcriptome index, you can pass those to Salmon and Kallisto, ask them to be quantified, and ask Salmon or Kallisto to give us back transcript raw counts as well as Bootstrap samples. You can import those to edgeR, and the only thing you need to do now is to divide your counts by the estimated mapping ambiguity over dispersion. And then you can proceed with your differential analysis. We did a very extensive simulation study to assess the performance of this count scaling approach. But here I'm going to present just a very brief overview of our simulations. So our simulation is divided in 4 main steps. And in the first step, we curated a set of transcripts for which we would generate expression levels. We used the ensemble-oriented gen code annotation for the mouse genome MM39, which we then gave those transcripts to R subread to generate FASTQ files, always comparing two groups and always generating 20 simulated data sets for every combinatorial scenario that we had. So we assessed different library sizes, different sample sizes, read protocols, so paired end versus single end reads, as well as read length, as well as the number of expressed genes or transcripts per gene. We then gave us those FASTQ files generated by R subread to Salmon and Kallisto to be quantified and get us back 100 Bootstrap samples for each simulation. And then we compared our count scaling approach not only with edgeR using the raw counts, but also with two commonly used methods for differential transcript analysis, which are sleuth and swish. Overall, what we saw was that by scaling down counts according to the mapping ambiguity over dispersion, edgeR provided the best power and the best false discovery rate control. And not only that, we saw that certain methods such as swish and even edgeR with raw counts, we had a hard time to control the number of false discoveries with a low number of replicates per group. But by scaling down those counts by the mapping ambiguity, we really could control the false discovery rate and provide better power. We also need null simulations for which we didn't have any type of differential expression between groups. And pretty much all the methods here are controlling as expected the type 1 error control in all the scenarios. Curiously, what we saw was that for sleuth, the distribution of null p-values was kind of asymmetric with lots and lots of transcripts with p-values near 1, which suggests that sleuth might be a bit over-conservative in our simulations. If you want to run your differential transcript expression analysis with edgeR, you can do that in less than 30 seconds. That's pretty efficient. So starting from Salmon counts, you can get your final list of GE transcripts in about 15 seconds or so. Whereas if you use other methods, that you need to wait a little longer. So like technology, many people here today, especially those from the Smythe lab, the Chen lab, the Rich lab, and the She lab, Andy and Gordon have been working together with me in the past year on this project, as well as Shui Dong and Matt Ritchie, who generated data for us to analyze, and Yang Liao and WEHI, who helped us along the way to simulate all these experiments. Also like to thank the Chan Zuckerberg Initiative for funding sources for this project, and happy to take questions from the audience.
Thanks, Pedro. Sorry, maybe I'll use this one. Questions for Pedro? We have a roving mic.
Thanks for the great talk. Can you hear me okay?
Yeah.
So I've got maybe two parts to this question. So the first is some genes have got more transcripts than others, and you showed that they had very different variance trends, especially the ones that only have one transcript versus if you had more, right? So I guess the first part of my question is, if you just look at the ranking of the genes that you end up identifying as changing, after you apply your method, do the genes that only have one transcript end up ranking higher than genes that have got multiple transcripts, even though they might still have changes?
So for genes that are expressing only one transcript, in general, what we are going to estimate the ambiguity will be very, very close to 1. So when we scale down those counts, pretty much has very little effect on those. When you talk about ranking, you're talking about ranking in regards to significance?
So I imagine when you apply this method and you rank things in terms of what's most differentially expressed versus not, if you apply it to raw, you're going to get some result. But then when you apply this method where you start moderating the variances, the ranking of those genes will start changing. I'm just wondering if the genes that have only one transcript end up ranking higher than those that have multiple. And if this is actually what we want to be happening, even though it's statistically correct, if it's actually what a biologist might want.
Yeah, I'm not sure. So if we compare the analysis with the count scaling and without count scaling, I would imagine that for genes that are expressing a single transcript, they would remain with the same significance. And but for genes that are overlapping, that has transcripts overlapping each other, those, we account for that variability. I'm not sure how the transcripts would move up and down with the count scaling. Sorry.
We can chat about it later. I'm actually really curious.